Let's study about photosynthesis in higher plants. All animals, including human beings, depend on plants for their food. Have you ever wondered from where plants get their food? Green plants, in fact, have to make or rather synthesize the food they need, and all other organisms depend on them for their needs. The green plants make or rather synthesize their food they need through the cycle through a process called photosynthesis and are therefore called autotrophs. You have already learned that autotrophic nutrition is found only in plants. All other organisms that depend on the green plants for their food are called heterotrophs. Green plants carry out photosynthesis and physicochemical process by which they use light energy to drive photosynthesis or the synthesis of organic compounds. Ultimately, all living organisms on earth depend on sunlight for energy. The use of energy from sunlight by plants doing photosynthesis is the primary source of food on earth. It is also responsible for the release of oxygen into the atmosphere by the green plants. Have you ever thought what would happen if there were no oxygen to breathe? This chapter focuses on the structure of photosynthetic machinery and the various reactions that transform light energy into chemical energy. The important thing here is we have to know what's happening in this para. So we are talking about the green plants. They make food with help of the light energy from the sun, and it undergoes more multi-step process called photosynthesis, and they produce food, and it's the only source of food on the earth, or the primary source of food on the earth. The main theme of this is the plants make up their own food, and so they are called as autotrophs, or the mode of nutrition they perform is called the autotrophic nutrients or the nutrition. So the organisms which depend upon the plants for food is known as the heterotrophs. As we know, the examples of heterotrophs or the uh, things which will come under the heterotrophs are human beings and animals. The next topic is, what do we know? Let us try to find out what we already know about photosynthesis. Some simple experiments you may have done in the earlier classes have shown that chlorophyll green pigments of the leaf, light and CO2 are required for photosynthesis to occur. You may have carried out experiments to look for starch formation in two leaves. A variegated leaf or a leaf was partially covered with black paper and exposure to light. On testing these leaves for presence of starch, it was clear that photosynthesis occurred only in the green parts of the leaf. Now let's consider a leaf. Okay, this is a leaf. So now what I'm doing is I'm gonna cover some part of the leaf with black paper or black cloths. So as we have learnt, the photosynthesis takes place only in the part where it have exposure to the light. That's this part. Why does the photosynthesis doesn't take place here? Because it doesn't have green pigment over here, but whereas here it has some green pigments. So by this way we can conclude that we can conclude that the photosynthesis takes place in all the green parts of the leaf due to the presence of light. So we can take the source of light from here over here. Okay, so the light comes from here to here, and thus it results in the process called photosynthesis. But where the part which was covered by the black cloths, the photosynthesis doesn't take place. Another experiment you may have carried out where a part of leaf is enclosed in a test tube containing KOH soaked in a cotton. Okay, why we are using KOH over here? Because KOH is a good absorber of CO2. That's why we are using KOH over here. While other half is exposure to air. The setup is then placed in a light for some time and tested for the presence of starch later in the two parts of the leaf. You must have found that the exposed part of the leaf tested positive. The exposed part we are getting the result as positive for the test for starch, while the portion that was in the test tube tested negative. This showed that CO2 was required for photosynthesis. For this reaction, mainly, I suggest you to consider a bell jar. Over here, we just imagine what I'm saying. So, imagine a plant, keep it inside this bell jar. Okay, so now we're gonna keep some KOH over here and keep another plant outside the bell jar. And I'm saying this just for an example, just consider this so you can get more clarity in this topic. Now expose this to sunlight. When you expose this to sunlight, the formation of starch only take place over here. It's, you know why? It's because the KOH is present inside the bell jar, so there would be no exchange of gases inside the bell jar. So the CO2 which was present inside the bell jar will be removed. So we can conclude that the presence of CO2 is needed for the process called photosynthesis. Now the very important topic over here is the yearly experiments. This yearly experiments is very important as we are getting more questions from this topic, and we have to be clear on the topic which scientist discovered what. It is interesting to learn about those simple experiments that led to gradual development in understanding of photosynthesis. The first scientist we are coming across is the Joseph Priestley, and over here I will highlight the important topics so you can note down the important keywords of this topic. Joseph Priestley in 1770 performed a series of experiments that revealed the essential role of air in the growth of green plants. This is very important because the Joseph Priestley, what did he do? He revealed the essential role of air in the growth of green plants. Priestley, you may recall, discovered oxygen. Who discovered oxygen? Priestley in 1774. This is also very important. Presley observed that candle burning in a closed space, a bell jar soon gets extinguished. Similarly, a mouse would soon suffocate in a closed place. He concluded that a burning candle or a animal that breathed the air both somehow damages the air. So yeah, from the earlier experiment, what we can conclude here? We can think that it's doing the burning candle or the animal is playing the role of KOH, as we have studied over here. We can compare and study for a good experience. But when he placed the mint plant in the same bell jar, he found that the mouse stayed alive and candle continued to burn. Presley hypothesized as follows: plants restore to the air whatever breathing animals and burning candles remove. So he says that something, the plant restores to the air whatever breathing animals and burning candles remove. So we know the answer. Yeah, you can comment down. Can you imagine how Presley would have conducted the experiment using a candle and a plant? Remember, he would need to rekindle the candle to test whether it burns only after a few days. How many different ways can you think of to light a candle without disturbing a setup? And this is a very interesting question. You can answer to this question in the comments. Using a similar setup as the one used by Presley, but placing it once in a dark. So this comes to the this is the second experiment over here. So first experiment. Okay, let's move on to the second experiment. So the same setup but placed once in a dark. So please note down this important keywords which I have highlighted over here for the first experiment. Okay, and let's move to the second experiment, that is the next experiment after Presley. We have to do the same setup but in the dark and once in a light. The name of the second scientist is nothing but Jan House, and the duration, the timeline is so and so. Show that sunlight is essential to the plant process that somehow purifies the air fouled by burning candles or breathing animals. Inzenhouse, in an elegant experiment with an aquatic plant, showed that in a bright sunlight, small bubbles were formed around the green parts, while in the dark they did not. So here the important keyword is that he used aquatic plants where in the bright light, and thus it resulted in the formation of small bubbles around the green parts, while in the dark they did not. We know in dark the photosynthesis doesn't take place. That is a reason for this. Later he identifies these bubbles to be of oxygen. Okay, the consider that this is an aquatic plant. As you have studied, the aquatic plants would look like this in a plant development lesson. Okay, as it is placed inside water, small bubbles will be seen. small bubbles will be seen around the green parts, and this bubbles are nothing but O2. This happens only when it gets exposure to the sun. Hence, he showed that it is only the green parts of the plants could release oxygen. Next, let's move on to the third experiment. It was not until about 1854 that Louis von Saches provided evidence for the production of glucose. Now comes the third important, very important experiment. Julius von Saches. What did he do? He provided evidence for the production of glucose when plants grow. The production of glucose. Here the keyword is the glucose. Glucose is usually stored as starch. You may get a question like, the glucose is usually stored as, or glucose is usually stored in which of the following forms? So the answer is starch. This latest study showed that the green substances in the plant's chlorophyll, that the green part is nothing but the chlorophyll, is located in special bodies. Where does the chlorophyll present? In the chloroplast. So for your understanding, inside the chloroplast, what is present? Chlorophyll is present. Chlorophyll is green in color, and they are responsible for the production of glucose. And how they are responsible? Because they store a form of glucose. That's nothing but they store starch in them. Within plant cells, he found that the green parts in plants is where glucose is made, and the green parts is nothing but the chloroplast, and the glucose is usually stored as starch. Next, let's move to the fourth experiment. Using the scientist name over here is T.W. Engelmann. The duration is 1843 to 1909. Using a prism, he split light into its spectral components and then eliminated a green algae. This is a very important question. Green algae, cladophora. Which algae? Cladophora. Green algae, cladophora, T.W. Engelmann. Placed a suspension of aerobic bacteria. The bacteria were used to detect the sites of O2 evolution. He observed that the bacteria accumulated mainly in the regions blue and red of the split spectrum. By this we can understand that the photosynthesis takes place at the high rate only in the blue and red light region. A first action of spectrum of photosynthesis was thus described. It resembles roughly the absorption spectra of the chlorophyll A and B. Okay, so I'll just conclude this. See, the name of the scientist is T.W. Engelmann. What did he do? He took a prism and then he took green algae. It's nothing but its name is cladophora, placed in a suspension of aerobic bacteria. Okay, when he kept all this green algae, cladophora in a suspension of aerobic bacteria, it's aerobic, so it needs oxygen. Here they can confuse you by giving anaerobic bacteria in the options. So the correct answer is the we have to take aerobic bacteria over here. The bacteria we use to detect the oocyte, it's because it is aerobic bacteria, so it gets accumulated only in the region of blue and red light. So it describes about how does the absorption of chlorophyll A and B take place, in which color what happens, they are discussing about it. By the middle of the 19th century, a key feature of plant photosynthesis were known, namely that plants could use light energy to make carbohydrates from CO2 and water. So by this way they are saying that carbohydrates can be made from CO2 and water with the help of light energy. Light energy is the main source, and what are the reactant? CO2, O2, H2O. Okay, what are the reactants? CO2 and H2O. And light is also used to produce what? To produce carbohydrates. And along with the carbohydrates we also get oxygen over here. The empirical equation representing the total process of photosynthesis for oxygen evolving organism was then understood as so and so. This is very important, but the empirical formula of this is nothing but CH2O. This is the empirical formula for what? Carbohydrates. Carbohydrates, namely it's nothing but glucose, mostly glucose. Glucose is a 6 carbon sugar, very important. A milestone contributed to the understanding of photosynthesis was that made by a microbiologist, Cornelius van Niel. So we can say it's Niel, it has Niel. And the duration of the scientist Cornelius van Niel, and the duration so and so, was based on his study of purple and green bacteria. Very important question. What kind of bacteria does Niel use? Niel used purple and green bacteria. Okay, and then what algae we have used in the above process? We have used green algae. In what reaction? To find what? To discover what? Okay, we have done that experiment to find the detection of the O2 in blue and red region, light region. Okay, based on his study of purple and green bacteria, demonstrated that the photosynthesis is essential a light dependent reaction. Okay, this is the fifth reaction. This is the fifth reaction. Photosynthesis is an essential light dependent reaction in which the hydrogen form a suitable oxidizable compound. So over here the hydrogen form a suitable oxidizable compound reduces CO2, carbon dioxide, to carbohydrates. So very important, CO2 to carbohydrates, it is whether it is reduced or oxidized. Very important, very important thing, because they can ask you that CO2 gets oxidized to carbohydrates or reduced to carbohydrates. They can give this in a statement form. Okay, so this is reduction. This is reduction, where over here oxidation. So reduction plus oxidation. What can we call this reaction as? We can call this reaction as you will have guessed, it's nothing but a redox reaction. It's nothing but a redox reaction. So, and another question over here, it's nothing but a photosynthesis is an example of reduction or oxidation or redox reaction. So the answer is that it's known as the redox reaction. In green plants, H2O is the hydrogen donor, and is oxidized to O2. Some organisms do not release O2 during photosynthesis. Some organisms do not release O2 during photosynthesis. When H2S instead is the hydrogen donor. So for the purple and green sulfur bacteria, the oxidation product is sulfur or the sulfate depending on the organism and not O2. So we are saying that oxygen is released or the sulfate is released. So it's because it depends upon why does this change. Okay, it changes because of the organism, the type of organism it belongs to. So over here we can see that the H2O is the hydrogen donor. H2O is the hydrogen donor. So from H2O some hydrogen will be removed. That's why O is formed. O is nothing but double bonds of O. Double bonds of O. O bonds results in the formation of O2. Actually, how many bonds are present between oxygen and oxygen? 2 bonds. Okay, 2 bonds. So, okay, how many bonds are present between oxygen and oxygen? 2 bonds, right? Okay. Hence, he inferred that the O2 evolved by the green plants comes from H2O and not from carbon dioxide. Very, very important. They can ask you as a statement. Okay. The O2 which was evolved by the green plants comes from H2O and not from carbon dioxide. Okay. This was later proved by using the radioisotopic technique. And this was proved by what technique? Radioisotopic technique. The correct equation that would represent the overall process of photosynthesis is therefore, this is very, very important reaction that we should never forget this. And this is nothing but 6CO2 plus 12H2O give rise to the carbohydrates formula or the glucose formula plus 6H2O plus 6CO2. As we have further studied that light is an important factor for photosynthesis. As we are discussing the chapter called photosynthesis over here. Photosynthesis, the synthesis which takes place with the help of photo. Photo means light. So as we are discussing about a chapter photosynthesis, right? Here we all have to notice that photo means light. Okay, light dependent reaction. Synthesis means reaction. So light dependent reaction. So facts about photosynthesis. Another fact is it's a kind of dash reaction. As I have already said, it's an redox reaction. Very important. Okay, where C6H12O6 represents glucose, the O2 released from water, as we have studied above. So nothing is new over here. We are just studying whatever we have studied in the before paras. This was proved using radioisotope technique. Know that this is not a single step, but this is a multi-step process. Okay, so that's why it's called as photosynthesis. Is photosynthesis a single step process? No, it's a multi-step process. Where does photosynthesis takes place? You would of course answer in the green leaf or in the chloroplast. Okay, where does photosynthesis takes place? Yeah, in green leaf or the green plants, as we have studied before, or in the chloroplast. What will be present inside the chloroplast? You have to answer. We have already studied that inside the chloroplast, what will be present? Chlorophyll will be present, and the chlorophyll is in green color substance. Yes. Yeah, let's continue. Based on what you have studied in the chapter 8, you are definitely right. Photosynthesis does take place in the green leaves or in the green parts, but it does so also in the other plants of the parts of the plants. Can you name some other plants where do you think photosynthesis may occur? Okay, what do you think about this? Yeah, let's see. You would recollect from previous unit that mesophyll cells in the leaves have large number of chloroplasts. Mesophyll cells in the chloroplasts, okay, that will mesophyll cells in the leaf will have large number of chloroplasts. Where does the large number of chloroplasts will be present? In the mesophyll cells in the leaves. Usually in the chloroplast align themselves along the walls of the mesophyll. If this is for example, if this is a mesophyll cell, okay, now what will be there in the walls? Chloroplast. Large chloroplast will be present in their walls of mesophyll. They are mesophyll, and this is chloroplast. We have studied the structures of chloroplast in chapter 8. Within the chloroplast, there is membranous system consisting of grana. So this is the chloroplast. Okay, it has membranous system. It has more membranous system, and it also has granas. So here we are calling this structure as grana. Okay, the stromal lamellae and the matrix stroma, there is a clear division of labor within the chloroplast. The membrane system is responsible for trapping the light energy, and also for the synthesis of ATP. So here we have to remember two things. The membranous system is responsible for what? It's responsible for trapping the light and also synthesis of ATP. What is the work of the membrane system? So here the work of the membrane system is nothing but to trap the light and synthesis the ATP. And very important, NADP also, NADPH. In stroma, enzymatic reactions synthesize sugar. In stroma, what is stroma actually? This green part, outer side of grana, but inner side of the inner side of the chloroplast is known as The stroma. In stroma, enzymatic reactions occur. Where does the enzymatic reactions occur? It occurs in the stroma. Enzymatic reactions occurs in the stroma. And why does this enzymatic reactions occur? It occurs for the synthesis of sugar, which in turn form starch. The enzymatic reactions occurs in the stroma. It happens for the synthesis of synthesizing the sugar. What will happen if we synthesize the sugar? We will get a starch. The former set of the reactions, since they are directly light are driven, are called light reactions. The former set, known as nothing but trapping of light and synthesis of ATP and NADP. This reaction is there, no? So this is called light reaction because it involves the characteristics, or it needs the help of light energy. That's why it's called as light reaction. And light reaction is also called as photochemical reaction. Photochemical reaction. The latter are not directly light driven but are dependent on the products of light. Which process depends upon the products of light? Which step? Yeah, the enzymatic step. Okay, the enzymatic step is called as the dark reactions. Okay, the production of ATP. See, the latter are not directly light dependent, are driven but dependent on the products of light reactions ATP and NADP. Hence, to distinguish the latter, they are called by convention as dark reactions, carbon reactions. So here we have to note one thing. It's nothing but light reaction is called as dash, that is, we know, photochemical reaction, then dark reaction is called as dash. Carbon reaction. Okay, very important to remember. However, this should not be construed to mean that they occur in darkness or that they are not light dependent. So by the way, they are telling that they are not, we should not consider them as light dependent, but actually they are not dependent on the light reaction, but they depend on the products of the light reaction. Okay, we have to be clear in that point. They are, we should not consider that the dark reaction takes place only during the dark times, but it can also take place in the light also. So we should not conclude that. Okay, now let's see this diagram, very important. This is the diagrammatic representation of an electron micrograph of section of chloroplast. So yeah, this is in chloroplast, as we all know. Okay, then this is the outer membrane, inner membrane, and this is known as the stromal lamellae. The stromal lamellae joins, helps for the jointment of granas, and yeah, the stack like arrangement is known as the grana, and This is known as stroma, and the small inclusion green color bodies are known as ribosomes, and this disc-like structures are called starch granules, and these are known as the lipid droplets. Let's move. How many types of pigments are involved in photosynthesis? Looking at plants, have you ever wondered why and how there are so many shades of green in their leaves, even in the same plant? So we come across plants which have variety of shades and colors, right? We can look for some answer to this question by trying to separate the leaf pigments of any green plants through paper chromatography. This chromatography is a technique which is used to distinguish the pigments which is present in the leaves. A chromatographic separation of the leaf pigments shows that the color that we see in the leaves is not due to a single pigment. So let's consider a leaf, okay, green color leaf. This is in the shade of green, okay. It can be dark green, light green, whatever it is, okay. This green shade, okay, it has some dark green or light green, whatever it is. It has these shades, right? Dark green and light green. It's not only because of a single pigment, it's because of varieties of pigments, okay. The pigments can be classified as so and so. They are chlorophyll A, chlorophyll B, xanthophyll, carotenoids, okay. Chlorophyll A, what color does a chlorophyll A pigment gives? It gives what? Bright or blue green, bright or blue green. In chromatogram, yellow green. So we can know this. Chlorophyll A gives bright or blue green or bluish green, okay. Chlorophyll B gives yellow green. Xanthophyll gives yellow, carotenoids gives yellow to orange. So let's recap. See, chlorophyll A gives what? Bright blue green, okay. And then chlorophyll B gives green. Then what do we have? Xanthophyll. It's from, okay, this one is green yellow, right? Green yellow. This one yellow. And then carotenoids have pigments ranging from yellow to orange, right? Yellow to yellow orange. Let us now see what roles various pigments play in photosynthesis. Pigments are the substances that have the ability to absorb. So if you want to note down the keywords of pigments, we can note down over here. Pigments are the substances that have an ability to absorb the light. Only the the thing which has the ability to absorb light are the pigments at a specific wavelength. Each pigment has specific wavelength so that it can absorb a specific magnitude of light. Can you guess which is the most abundant plant pigment in the world? Do you know? Okay, let us study the graph showing the ability of chlorophyll A pigment to observe lights of different wavelengths. Of course, you are familiar with the wavelength of the visible spectrum of light as well as the VGR: violet, indigo, blue, green, yellow, and orange, red. Okay, from figure 11.3a, can you determine the wavelength of color at which chlorophyll chlorophyll A shows the maximum absorption? Does it show another absorption peak at any other wavelength? If yes, which one? Now look at the figure. Okay, we'll see this, and by seeing this, what we can conclude is that, see over here, um, this is the chlorophyll A's peak 1 and 2. It has two peaks, this one and this one. So by this way, we can conclude that it has two peaks, and this one, this wavelength they have given, right? It's applicable for this all these places straight. Okay, so with this wavelength we can match with the name of the colors, and we can check. By checking it, we'll get an information that chlorophyll A, right? Chlorophyll A will get maximum peak at two colors, where it's nothing but blue and red. Hope you understand. Now look at the figure 11.3 showing the wavelength at which the maximum photosynthesis occur in a plant. Can you see that? Wavelengths at which there is a maximum absorption chlorophyll A. Okay, now only we have studied about it in the blue and red region. Also show higher rate of photosynthesis, hence we can conclude that chlorophyll A is the chief pigment. So this is a very important question. Which is the chief pigment? Chlorophyll A is the chief pigment associated with the photosynthesis. But by looking at the figure 11.3c, can you say that there is a complete 1-to-1 overlap between the absorption spectrum of the chlorophyll A and the action spectrum of the photosynthesis? See, action spectrum. When is the action spectrum and the absorption spectrum of the chlorophyll A coincide? When is the action spectrum? Okay, when is the action spectrum and the absorption coincide? When is the action and absorption coincide? It coincides at which point? It coincides at this points, these points. Okay, and as we check their wavelengths, okay, as we check their wavelengths by using these wavelengths, we can conclude at which color the photosynthesis rate is higher. Okay, these graphs together show that most of the photosynthesis takes place in a blue and red region. As we can compare and study, we get an information that in the place of blue and red region, the rate of photosynthesis is higher. Some photosynthesis does take place at the other wavelength of the visible spectrum. In the other wavelength of the visible spectrum also the photosynthesis takes place, but at blue and red region it takes place at the most. Okay, more fastly. Okay, let us see how this happens. Though chlorophyll A, wait a minute. Though chlorophyll is major pigment responsible for trapping the light, other thylakoid pigments like chlorophyll A, chlorophyll B, xanthophylls, carotenoids, which are called as accessory pigments. Next important thing is, what are accessory pigments? Accessory pigments are nothing but the chlorophyll B, xanthophylls, carotenoids. That is nothing but the pigments except chlorophyll A. Okay, also observe light and transfer the energy to chlorophyll A. Indeed, they are not enable a wider range of wavelength of incoming to be utilized for photosynthesis, but also to protect chlorophyll A from photooxidation. Okay, also observe, see, they also observe the accessory pigments. The accessory pigments observe the light, they also observe light, but they transfer the energy to chlorophyll A. Why they transfer the energy to chlorophyll A? Because chlorophyll A is called as chief pigment. See, over here they have given the information. Chlorophyll A is the chief pigment, very important, and it also prevents the photooxidation of chlorophyll A. Next, what is light reaction? Light reactions of the photochemical phase include light absorption. The light reaction refers to the light absorption, water splitting, oxygen release, and the formation of high energy chemical intermediates. See, very important thing, a very important question you can get here, that what is the main step in the light reaction? Water splitting, oxygen release, light absorption, and the formation of high energy chemical intermediates, that is nothing but ATP, NADP. First step is light absorption. By the light which was observed, the water will be getting splitted. As the water gets splitted, the two hydrogens will go out and will get oxygen, right? Then it will result in the release of O2 molecule, that is nothing but oxygen release or O2 release, and the formation of high energy. As all this happens, we will get an high energy intermediates, that is nothing but the ATP and NADP. As I hope you all know the full form of NADPH and ATP. I request you to comment down this. Okay, their full form of ATP and NADPH. Several protein complexes are involved in the process. The pigments are organized into two discrete photochemicals. See, the pigments, we are talking about the pigments, right? We are talking about the chlorophyll B and xanthophylls, right? Carotenoids. All these pigments are organized into two discrete photochemical light harvesting complexes within the PS1, PS2. So there are two LHC, or the light harvesting complex. Let's think this is a complex and this one is another complex. Here we will have some kind of some set of pigments. Here some set of pigments will be present, and over here some set of pigments will be present, and this one is known as PS1, PS2, and this one is PS1. And you may wonder why I'm writing PS1 first and you may wonder why I'm writing PS2 first and PS1 second, and in the further you can later you can get the answer for this. These are named in sequence of their discovery. As I said, here is the point, here is the answer. They are named in the sequence of their discovery and not in the sequence of which they function. So PS2 means it is discovered after the discovery of PS1. That's why it's named as PS2 and it is named as PS1. The LHC are made up of hundreds of pigments. Okay, what is LHC? Light harvesting complex. Okay, it is made up of hundreds of pigments molecules bound to a protein. Each photosynthesis has all the pigments except one molecule of chlorophyll A. Each photosystem has all the pigments. Okay, both the PS1 and PS2 has all the pigments except A. Okay, forming a light harvesting system, also known as antenna. antennae or antenna. So in antenna, what will be missing? Yeah, correct. It's nothing but a chlorophyll A will be missing in the antenna. These pigments help to make photosynthesis more efficient by observing the different wavelengths of light. Single chlorophyll A molecule form reaction center. So in we have two things over here, antenna and reaction center. Okay, antenna, in antenna it excludes chlorophyll A, but in reaction center, what is known as the reaction center? Only the chlorophyll A is known as the reaction center. The reaction center is different in both photosystems. PS1, the reaction center is chlorophyll A and has a maximum, okay, PS1, in PS1 only the reaction center will be present. And see, in PS1 the reaction center is chlorophyll A and has maximum absorption peak of 700 nanometer. You know what is nanometer? Yeah, you can comment down. Hence is called P700 because it observes the wavelength of 700. While in PS2 it has the absorption maxima at 680 nanometer. In PS2 it has the maximum absorption peak around 680 nanometer. Next, another question. Chlorophyll A will be present in PS1. Yes. Now let's study about the ETS, electron transport system. In photosystem 2, the reaction center chlorophyll A, as we all know that in PS2 the reaction center is chlorophyll A and absorbs 680 nanometer. Is it so? Yes. Sorry, for a clarification, there will be reaction center in PS1 and PS2. Okay, in both the chlorophyll A will be present, but in PS1 only the chlorophyll A will observe the absorption peak at 700 nanometer. In PS1 only it observes 700 nanometer. Wavelength of red light causing electrons to become excited and jump into an orbit farther from the atomic nucleus. These electrons are picked up by the electron acceptor, which passes them to an electron transport system. Yeah, I have to teach you this very important and exciting thing. So this is nucleus. Okay, this is nucleus. What is the charge of nucleus? Neutral, zero charge. Next we have the orbits, right? What orbits? The orbits in which electrons will be present. Okay, this is the outer orbit, and this is near to the nucleus. Okay, now consider here some electrons are there. Consider some electrons over here, here. Now what happens is some wavelength of light, for example 680 nanometer, comes from sun. Okay, from sun we are getting some wavelength of light. So this wavelength of light reaches the nucleus, which is inside the it reaches the electron which is inside the atom, and due to which it gets excited because some energy flow takes place. Energy flow takes place over here, so energy gets transformed from this to here, and then the electron gets excited and it moves further away, and it goes to the farther orbital. Farther means which has more distance from the nucleus. So it gets energy from the incident light, okay, and gets excited and moves farther away to the outer orbital. As it moves to the outer orbital, an electron acceptor comes. Okay, an electron acceptor comes over here. As some electron acceptor pass through this, as they have shown here, it goes inside the electron transport system and hence goes to the second light harvesting system, and again the same process occurs, that is nothing but the electron acceptor. It goes and bind to the electron acceptor, and then the process again results in the production of NADPH. And in the first process, ADP is converted into ATP in the presence of inorganic phosphate. Actually, now you all need to know the full form or the abbreviation of ATP. What is ATP? Adenosine triphosphate. Okay. Yeah. Passes them to electron transport system consisting of cytochrome. These are cytochromes, which is shown over here. Okay. Here. This movement of electron is downhill as it moves. See carefully. This reaction, okay, from here up then to down. This looks as downhill. So movement of electrons is downhill in terms of oxidation, reduction or redox potential scale. What is redox potential scale? Oxidation reduction in the downhill is known as the redox potential. The electron are not used up as they pass through the electron transport system. As the electrons, the electrons are accepted by the electron transport. Electron acceptor, right, and then it gets transferred into the ETS known as electron transport system, and in the system no electrons are used. Okay, but pass on to the pigments of the PS1. Where is PS1? Over here. Simultaneously, electrons in the reaction center PS1 are also excited, where they receive the red light of wavelength 700. Because here what is present in PS1? In PS1 what is present? We have chlorophyll. We have chlorophyll A, right? That's why. Because that chlorophyll A will receive the light of 700 nanometer and are transferred to another acceptor molecule that has greater redox potential. The thing which has greater redox potential can accept electrons. Okay, you have to keep in your mind. These electrons are then moved downhill again. So again the same process takes place, again up and then down, and this looks like this, right? Like this. So we can call this scheme as the Z scheme also. These electrons are all moved downhill again, this time to a molecule of energy rich NADPH. The addition of these electrons reduces NADPH to electrons reduces NADP plus to NADPH plus H plus. This whole scheme of the transfer of electrons starts from PS2 uphill to the acceptor. So we all need to know that where, like, how, what is the process happens here over here. The process starts from PS1 and then goes to PS2. And why it got the name PS1 and PS2 as it's given, okay, already. Why uphill? Where is uphill? Where is downhill? And which is the acceptor of the electrons? Everything you have to study. Down the electron transport chain to PS1, excitation of the electrons, transfer to another acceptor, and finally downhill to NADP plus, reducing to NADP H plus. So here we have to know that which is the reduced form and which is the oxidized form. NADP H plus is the reduced form, and NADP plus is the oxidized form, and this is the reduced form. It's called Z scheme. Due to its characteristic shape, this shape is formed when all the carriers are placed in a sequence on a redox potential. The shape, how do we get this shape? We get this shape when all the electron carriers are placed in a sequence on a redox potential scale. We all need to know the splitting of water. Okay, you would ask then, how does PS2 supply electrons continuously? See, over here, if some amount of electron gets excited and goes to the electron acceptor and all this process happens, what will happen over here? Here some positive gradient will be formed, right? So to compensate that, we need electrons, right? How does the replenishment of the electrons takes place in this reaction? The electrons that were moved from PS2 must be replaced. Okay, this is achieved by the electrons available due to the splitting of water, and this will be, see, very important key point, by the splitting of water. Okay, the splitting of water is associated with the PS2. Water is split into 2H plus and one nascent oxygen, or the natural form of oxygen and electrons. Now let's say gives 2H plus and one nascent oxygen and then electron. This creates oxygen, one of the net products of photosynthesis. The electrons needed to replace those removed from photosystem 1 are provided by photosystem 2. The electrons needed to replace those removed from PS1, okay, are provided by PS2. So for conclusion, the electron from this one will be replaced by the water, okay, and who will give electrons for this? PS2 will give for this. Keep in your mind, PS2 will give. Not clear? You can try this. Okay, we need to, this is the reaction that is given over here, and you may get some doubt. Some people may get doubt that if an H2O molecule is split, how much H plus we get? 2H plus. Yes, but why here 4H plus? Yeah, you are right. This is 1 mole. Okay, this is 1 mole of water, 1 mole or 1 molecule of water. Over here it is 2 moles. Here 1 mole of water, here 2 moles of water. That is the difference. If okay, they can also ask this kind of question: if 2 moles of water is taken, how many protons do we get, or the number of electrons we get? Okay, we need to emphasize here that the water splitting complex is associated with the PS2, which itself is physically located on the inner side of the membrane of thylakoid. Very important, we have to know the location where does this takes place, and over here the PS2 is located in the inner side of the thylakoid membrane. Then where are the protons and O2 formed likely to be released in the lumen or on the outer side of the membrane, and about it we will be studying in the chemiosmosis. Okay, now let's move to cyclic and non-cyclic phosphorylation. What is photophosphorylation? Addition of phosphate in the presence of light, known as photophosphorylation. If this reaction, what I have said, takes place in a cyclic manner or in a non-cyclic manner, okay, that defines the the topic. Okay, living organisms have the capability of extracting energy from oxidizable substances. Living organisms gets energy from what? Gets energy from the oxidizable substance, the substance which has the ability to oxidize. Okay, and store in the form of bond energy. What bond energy? You all, I hope you all know the formula of glucose. It's nothing but C6H12O6, right? And over this, where does the bond energy will be, like the bond gives, which bond gives energy? Okay, which bond gives energy? The C to C bond is present, right? And this bond will provide energy. Special substances like ATP carrying this energy in the chemical bonds. The process through which the special substance, the ATP, or the, like, from this bond only we get ATP. You will study further more about this. Okay, ATP is synthesized by cells in mitochondria and chloroplast. Where does the ATP is synthesized? ATP is synthesized in the mitochondria and chloroplast, and its name is phosphorylation. Where does the phosphorylation occurs? In mitochondria and chloroplast. Photophosphorylation is the synthesis of ATP from ADP and inorganic phosphate, which we can see from the heading. We can understand more from the heading itself. Okay, in the presence of light, because it's photo means it's a photophosphorylation, so it takes place in the presence of light. When the two photo system works in a series, that is first PS2 and then PS1, this process is called non-cyclic because it's cycle. See, it takes place, the reaction takes place, first PS2 happens, then PS1. This is a cycle, and it gains some electrons from H2O, water splitting, and this gives some electrons. All these are cyclic. Okay, the two photosystems are connected through an electron transport chain, as seen earlier in the Z scheme. Both ATP and NADPH are synthesized by this kind of electron flow. But when only PS1 is functional, so now we are coming to the cyclic phosphorylation. The electrons is circulated within the photosystem and the phosphorylation occurs. So in this, the cyclic phosphorylation occurs only in the PS1. Okay, and cyclic flow of electrons, a possible location where this could happen in the stroma lamellae. Stroma lamellae. In stroma lamellae, PS1 will be present, right? Where does the PS2 will be present? It's here, right? In the inner membrane of the thylakoid, PS2 is present. Inner membrane of the thylakoid, PS2 is present. And where does PS1 is present? PS1 is present in the stroma lamellae. While the member of the lamellae of the grana have both PS1 and PS2, the stromal lamellae members lack PS2 as well as NADP reductase enzyme. Well, okay, I think you can understand. Well, the membrane or the lamellae of the grana have both the things, there is both the PS1 and PS2. The stromal lamellae member lack PS2 as well as NADP reductase. Why? Because the work of PS2 and the PS1 are not correlated here, because PS2 is absent. So it lacks NADP reductase. The excited electron does not pass to ADP plus, but is cycled back to PS1 complex through the electron transport chain. The cyclic flow hence result in the synthesis of ATP but not NADPH. The cyclic photophosphorylation also occurs when the light of wavelength beyond 680 nanometer are available for excitation. So, with the help of both the PS1 and PS2, we can do this. But without the PS2, we cannot produce, we cannot give NADPH, okay, because it lacks NADPH reductase enzyme, right? Then this is one of the very, very important topic, chemiosmosis. And then let's see, chemiosmotic hypothesis. Let us now try and understand how actual ATP is synthesized in the chloroplast. The chemiosmotic hypothesis has been put forward to explain the mechanism like the respiration in photosynthesis too. ATP is synthesized linked to the development of proton gradient. So, as we are studying about the chemiosmotic hypothesis, we have to know the proton gradient across the we will be studying about the proton gradient. Why does this proton gradient come? It comes because we have to do the ATP synthesis. The proton gradient is important for the ATP synthesis. Okay, this time these are the members of the thylakoid. There is one difference, though. Here the proton accumulation is towards the inner side of the membrane, that is in the lumen. In respiration, proton accumulation in the intermembrane of the space in the mitochondria where the electrons move through the ETS. So here can you understand what they are coming to say? In respiration, what takes place is the proton accumulation takes place in the intermembrane space in the mitochondria. That in the respiration in mitochondria takes place in the intermembrane, but whereas here over the chloroplast, it takes place in the inside of the membrane. Here intermembrane, here inside of the membrane. Inside of the membrane, that is in the lumen. This happens in the lumen, but whereas in mitochondria it takes place in the intermembrane. Let us understand what causes the proton gradient across the membrane. We need to consider again the process that takes place during the activation of the electrons and the transport to determine the steps that causes a proton gradient to develop. So what are the causes of the development of the proton gradient? We are going to discuss now. Since the splitting of water molecule takes place on the inner side of the membrane, the protons or hydrogen ions that are produced by the splitting of water accumulate within the lumen of thylakoid. Why it takes place inside the lumen of the thylakoid? Because the water splitting takes place in the inner membrane, inner side of the membrane. As the electrons move through the photosystems, the proton are transported across the membrane. So we are just talking about the electrons in the PS1 and PS2 LHC light harvesting complex, but we did not talk about the four proton which was released during the splitting of splitting of water reaction. This happens because the primary acceptor of the electron, which is located towards the outer side of the membrane, transfers the electron not to an electron carrier but to an H carrier. So here E acceptor we have studied, right? This E acceptor means what? That and this has some connections. Okay, it transfers to an electron not to an electron carrier but to an H carrier. This is it is an H carrier. Okay, hence these molecules removes proton from the stroma while transporting the electrons. So these molecules remove the proton from the stroma while transporting the electron. When these molecules pass on its electron to an electron carrier when these molecules pass the electrons to an electron carrier on the inner membrane of the inner side of the membrane, the proton is released into the inner side of the lumen of the membrane. The NADP reductase enzyme is located on the stroma instead of the membrane. Where does the NADP reductase enzyme is present? It is present in the stroma side of the membrane. Along the electrons that comes from the acceptor of the electrons of PS1, the proton are necessary for the reduction of NADP plus to NADPH2 NADP plus to NADPH plus H plus. So here we are saying that electrons and protons both are important for this reaction. These protons are also removed from the stroma. Hence, these 3 points are very, very important, and I'll summarize this for you again. Hence, within the chloroplast, proton in the stroma decrease. This is the chloroplast, the stroma, and these will be the granas, right, and thylakoids, and protons in the stroma will decrease in number, while in the lumen there are accumulation of protons. Okay, so by this way we can say that in lumens more H plus will be there, but whereas in the stroma less number of H plus will be there, because the water splitting takes place in the inner membrane. This creates a proton gradient across the thylakoid membrane, as well as the measurable decrease in the pH in the lumen. So the pH will decrease as the H plus concentration increases. Why are we so interested in the proton gradient? This gradient is important because the breakdown of the gradient that leads to the synthesis of ATP. The gradient is broken down due to the movement of the protons across the membrane to the stroma through the transmembrane. So now now from lumen to stroma, the membrane should be broken down through what? Through transmembrane. Channel of F0 of the ATP synthase. The ATP synthase enzyme consists of two parts. That is nothing but CF0 is embedded in the thylakoid membrane and forms the transmembrane. What forms the transmembrane? CF0 forms the transmembrane channel that carries out the facilitated diffusion of the proton across the membrane. The other portion is called the CF1 and protrudes on the outer surface of the thylakoid membrane on the side that faces out the stroma. See, this is stroma. This is stroma. So here membrane is there. Here membrane is there. Here F0 is there. Oh, sorry, FO is there. Here F1 is there. The breakdown of the gradient provides enough energy to cause conformational changes in the CF1 particle of the ATP synthase, which makes an enzyme synthesize several molecules of energy packed ATP. Chemiosmosis requires a membrane. Okay, very important question over here we will get. Chemiosmosis requires what? Chemiosmosis requires a membrane, proton pump, proton gradient, ATP synthase. Okay, 1, 2 and 3, 4. 4 things are required for chemiosmosis. What are the 3? What are they? The chemiosmosis requires membrane, proton pump, proton gradient and ATP synthase. Membrane, proton pump, proton gradient, ATP synthase. Energy is used to pump the protons across the membrane to create a gradient. High concentration of the proton within the thylakoid lumen. ATP synthase has a channel that allow the diffusion of proton back across the membrane. The ATP synthase has a channel that allow the diffusion of the proton back across the membrane. This releases the enough energy to activate the ATP synthase enzyme that catalyzes the formation of ATP along with the NADPH produced by the movement of the electrons. The ATP will be used immediately in the biosynthetic reaction. So here in the reaction takes place in the stroma, responsible for the fixing CO2 and synthesis of sugar. So in the last point, they are coming to say that along with the NADPH which was produced in the before process which we have studied through LHC, light harvesting system. In LHC NADPH is produced, right? So along with the NADPH produced, this ATP which was produced by the chemiosmosis hypothesis, or these ATP and NADPH will be used in the biosynthetic reaction taking place in the stroma. Where does the biosynthesis reaction takes place? In the stroma. So it is responsible for the fixing of CO2 and synthesis of sugar. That is nothing but the photosynthesis. So 11.7 we'll see in the next part. We'll see in the part 2. Thank you.